The mammalian startle response is a biological reflex that initiates in the nervous system before conscious thought can intervene. Shock is a pre-effective physical collision. An acute environmental impact strikes the brainstem milliseconds before the mind can generate a narrative or recognize fear. In a healthy system, this rapid influx of sensory data flows smoothly through the neural architecture. The mammal orients to the stimulus, assesses the threat, and returns to its physiological baseline. In a traumatized system, the sheer force of the stimulus overwhelms the brain's processing capacity. The network loses its ability to correctly classify intense auditory or physical inputs as harmless. This failure alters the physical state of the network. The smooth pathways of communication transform into a state of vibrating, static tension, essentially fraying the neural connections. Trauma begins as a mechanical malfunction in the body's oldest, most primitive survival hardware. Deep within the pons lies the locus ceruleus. This dense cluster of neurons acts as the brain's master survival switch. This is the blue spot in third gear. In this state, the network fires noradrenaline in quick, burst-like successions. Noradrenaline is a chemical messenger that heightens alertness and prepares the body for immediate action. This massive chemical surge serves as a vital wake-up call. To ensure immediate reaction, the noradrenaline deliberately forces the prefrontal cortex, the area responsible for judgment and complex thought, offline. Bypassing higher-order thinking allows the brain to instantaneously engage the amygdala and sensory motor networks. The body initiates an automatic defense before the conscious mind can process the event. Chronic trauma occurs when the external threat vanishes, but the locus ceruleus fails to reset. This leaves the sensory motor networks locked in a state of hypervigilance. A traumatized mammal is physically trapped in a disembodied suspension because its chemical survival switch is jammed in the on position. Biological shock is not a single monolithic event. It fractionates into specific neurobiological cascades depending on the nature of the environmental trigger. Impact shock is the biological response to an acute, sudden threat, such as an assault or immediate physical danger. Implosive shock arises from a different environment. This pathway activates in response to prolonged, inescapable neglect or isolation during early development. This cascade moves from the giant cell nucleus of the medulla into the lateral parabrachial nucleus and the periaqueductal gray, eventually reaching the hypothalamus. These distinct physical routes produce radically different somatic sensations. Impact shock often manifests as a piercing, icy stab, while implosive shock creates an inwardly compressing, hollow pressure inside the head. The brain maps emotional abandonment and physical violence through separate neurological highways. Severe neglect leaves a physical scar on the nervous system that is as measurable as a physical blow. Vestibular shock is triggered by sudden relational rejections or a literal fall into darkness. In this form, the shock routes aberrantly through the brainstem's vestibular nuclei and into the flocculonodular lobe of the cerebellum. This specific pathway creates the profound somatic sensation of tumbling, being wrong-footed, and feeling utterly lost in physical space. In the state of horror, extreme pre-affective shock physically merges with deep revulsion. This sensation originates in the lower medulla, specifically within the area postrema and the nucleus tractus solitarius. These specific structures evolve to detect circulating physical toxins in the bloodstream and trigger vomiting to expel them. The traumatized brain repurposes this ancient toxic defense system to process horrific visual or psychological stimuli. To manage unbearable psychological realities, the brain recruits its oldest hardware, turning moral revulsion into literal biological nausea. Aloneness and social abandonment are profound, measurable biological stressors. Deep in the brainstem, the dorsal raphe nucleus monitors inescapable stress. This nucleus triggers the release of interleukin-1 beta. This pro-inflammatory cytokine is a signaling protein that actively drives self-imposed social withdrawal to limit the spread of perceived disease. Longitudinal clinical data shows that childhood social isolation is robustly correlated with elevated systemic inflammatory markers decades later in adulthood. Researchers specifically track elevated levels of SUPAR. SUPAR is the soluble form of a blood protein receptor that serves as a sensitive marker for systemic chronic inflammation. When pre-effective shock cannot be integrated by the brain, it spills over, mutating from a localized neural event into a systemic pro-inflammatory disease that degrades the entire body. During periods of intense stress, the brain may enter a state of dissociation. It generates as-if states in the insular cortex, simulating the physical feelings of trauma even when the body is safe. The insular cortex is a deep brain region that facilitates the conscious awareness of internal bodily states. This keeps the traumatized mammal trapped in an endless, disembodied loop, completely unable to recognize that time has passed. This anatomical diagram shows the cerebellum, which acts as the brain's Bayesian predictive engine. The Bayesian brain theory suggests the brain operates as a prediction machine, constantly using past experiences to update its internal models of reality. During traumatic memory retrieval, the connectivity between the brainstem and the cerebellum becomes massively amplified, flooding the predictive engine with panic signals. This amplification causes a secondary effect. The crucial connections between the cerebellum and the higher cerebral cortex are simultaneously weakened. As this connectivity fails, the brain's internal predictive models become perfectly rigid, unable to intake new context. PTSD stems from an impairment of the brain sequencer. The mind freezes inside a shattered prediction of a dangerous world. To properly break the loop of disembodiment, clinical interventions must focus on restoring the rhythm of the brain sequencer. Healing engages the downward cortico-pontocerebellar tract. This tract delivers slowed-down sequences to the cerebellum, feeding the engine manageable data instead of overwhelming shock. This completes the upward feedback loop, the cerebello-thalamo-cortical tract. This closed-loop sequencing allows the cerebellum to contextualize the brainstem's alarms, finally updating the rigid survival models with new data. As the sequencing continues, the chaotic connections of the insula and brainstem begin to knit back together. Neurological recovery relies on slowing down and sequencing raw data to reorganize the pathology of shock. Through patient-sequenced integration, the raw neurological pathways of survival can be re-smoothed into a resilient, interconnected architecture.